This podcast is brought to you by LMU Munich.
Hi everyone, can you hear me okay? Alright, I'll go ahead and get started. So I'm going to talk about remote controlling fish with Python. So this is a little bit of a last minute talk. I didn't know I was going to give it until last night. So yeah, it'll be fun, very spontaneous. My name's Joe and I'm from the Biolab at the Max Planck Institute for Neurobiology, which is just outside of Munich. So I would argue that you could consider the brain as one of the most interesting computer programs possible. Except it's not a computer, but it processes its environment to adaptively control multiple complex output sequences. And you have a setup where there's some inputs, there's some processing, and then there's some outputs. So that actually looks a lot like a pretty simple scripting. Unfortunately, it's not so simple. Have you ever gone into someone else's project and tried to read it and realized that the language is terrible, even worse than Perl, and the hardware is really weird and quite variable? So I'd like to introduce the larval zebrafish, which is a small fish. At this age, it's about 1 centimeter long. It's transparent, so we have great access to the brain, and we understand a lot about the genetics, so we can do some pretty interesting modifications, which I'll mention later. And it has a variety of interesting behaviors. So in this case, the fish is hunting down a little paramecia as food. So there's an interesting level of complexity, enough to be challenging, but also to be possibly solvable. So the setup that we use for experimentation looks something like this. So to keep the fish in place so we can actually look at it while it's doing the experiment, we embed it in some soft gel. And then we can put a display up in front of the fish and show some stimuli using OpenGL and some nice Python programs like CyclePy and VisionEgg. We can take an infrared camera and some infrared LEDs and then record the fish in infrared and then track it using OpenCV. So here's the fish responding to some stimuli shown on the screen, and it swims. And you can see these black spots along the tail are tracking. And then we can automatically extract bouts so that at this age the larval zebrafish swims in separate bouts as opposed to continuous motions. And to talk a little bit real quick about how the tracking works, my first thought was, okay, do some thresholding. That shouldn't be so hard, right? And not quite so easy. So to have thresholding that would work on any particular setup with a certain set of lighting and a certain lens and a certain camera, okay, not very hard. But then on a different setup or fish with different pigmentation, all of a sudden it would break and we would adjust the threshold and then it would break somewhere else. And what turned out to work a lot better was that to just try to track the midline of the fish. Because anyways, that's the most important part, and we want this to be very fast in case we want to do this real time. So the user starts out by just clicking a point on the tail and giving it a direction, and then we take a slice through the tail and we look at the data there, and of course the tail is a different color than the background, so we can find the midpoint, and we just do this repeatedly, each time adapting it to the direction of the tail, and at the end we get a midline through the tail that we can use to quantify the behavior. So real quick side note about some common languages in science. So unfortunately, MATLAB and LabVIEW are very, very common, and you find surprisingly often people doing reasonably involved data analysis in Excel with lots of copy and pasting, which is quite unfortunate. And there's also a reasonable amount of statistical languages like R used. Python's a lot better. I don't think I have to convince any of you about that. It's popular, and it's getting increasingly popular, which is really wonderful. And version control is super useful in science because unlike in some industries where you have a script and it doesn't change very often, in science people are constantly trying to tweak things and improve the analyses. Unfortunately, version control right now is a little bit tricky for many beginning programmers or biologists, people coming from other fields. So back to the neural circuits. So we really want to understand the processing, but how do you set a breakpoint in a fish? Well, what do you want to do? The first thing you want to do is observe the internal state, right? Like at least a print function that tells you what some variable is doing. And it'd be nice if you also could do some manipulation. So how do we look at the activity? Well, luckily we have some really nice genetic access, and there's some cool tools that have been developed. Specifically what I'm talking about today is a calcium detecting activity sensor. So it's this genetically modified protein that's put together from a mix of a few other proteins that effectively is something that fluorescence, so it glows when you shine a blue light on it, and also something calcium sensitive. And together they make something that glows in a calcium sensitive way, and it happens that when neurons fire, the amount of free calcium goes up, and then you get some illumination. And also we have some genetic control systems, so rather than just putting this everywhere in the fish, we can selectively pick groups of neurons or certain brain regions to put this into and just look at the specific subcircuits. So this is the fish brain, specifically the nucleus of the medial longitudinal fasciculus, which is a terrible name, and I won't say it again for the rest of the talk, but that's the region I'm looking in. And here's the fish tail. So you can see that as the fish swims, the brain lights up. And there's quite a reasonable amount of pre-processing for this. Some of it's fairly easy, like doing some motion correction, some filtering. One of the hardest parts is actually handling the metadata and image formats, some of which are proprietary or buggy. So there's quite some challenges there. So I also promised to talk a little bit about lasers. So this is how fluorescence works. Essentially, you have some energy, you excite a molecule to a higher state, it drops down to a slightly different state, and a different color of light's emitted back out. And for normal imaging, you have a certain amount of focus, just like you would with a camera. But unlike for a camera where you usually want a bigger depth of field, for microscopy you want a very, very small depth of field. So you can look at just a small section of the fish at a time with very nice resolution and not be looking at things out of focus and won't give you a clear picture. And so a cool trick we can use is called two-photon imaging, or uses the two-photon effect. So effectively what we do is instead of one high-energy blue photon, we have two low-energy infrared photons. And if these hit the same molecule at about the same time, then you have a similar effect. But the cool thing is now we introduced a nonlinearity. So instead of being related to the first order, it's now the second order, so it makes the focus much more narrow. This also improves the penetration, and the laser is now invisible. And the lasers we use are actually really cool. So because this is now second order, the timing of the laser matters, and so it only works practically if you compress the laser energy into very short pulses, so only 140 femtoseconds, and then 80 million pulses per second. And the lasers are also tunable in wavelength, which is pretty unusual. All right, so now we can observe the internal state of the fish and see what the circuits are doing during behavior. But it would be really great to be able to manipulate the state. Fortunately, we have some cool tools for that as well. So there's genetically encoded light-sensitive channels that are actually very similar in structure to what you'd find in an eye, where light comes in and it activates a neuron. So we can use a laser, shine it onto neurons expressing these channels, and then they let some ions through that drives activity in these neurons. So here's remote control of a fish. So at the top there's an optic fiber that's coming down to shoot a laser into the fish's brain. And once the video starts, you'll see that we move the optic fiber back and forth once it's activated, and the fish moves its tail back and forth. And the optic fiber is moved from the left side to the right side, and you can see with a slight latency, the fish's tail moves to match the optic fiber. And if we map this out a little bit more thoroughly, we can see that if you activate on the right side in the right spot, you get some swimming and tail deflections. If you activate on the left side in the opposite of that same spot, you get to the other side, and if you activate some of these other neurons, then nothing happens, which is really cool, but that's still very coarse. So you're activating maybe hundreds of different neurons, and you don't want to know what 100 different neurons do. You want to know what just this neuron does or that neuron does. So how can you get more specific? So with an optic fiber, you don't have very good control. You can use a projector, actually very, very similar to what's right here, but that's only in 2 dimensions, and the brain is not a 2-dimensional structure. It's 3 dimensions. So how do you shape the light into a 3-dimensional pattern with good resolution? Well, the answer is actually holography, which sounds like something out of sci-fi, but it exists and it works and it's very cool. So let me explain it to you. So light is a wave, and if you take a flat wavefront and you focus it through a lens, you'll get a point. But if you introduce a slight offset in that wavefront, so you could think of this actually as a time shift, so that you just delay the wavefront a little bit in certain places. Then after it goes through the lens and focuses, it's only temporally coherent in a different spot. So by changing the timing of this light wavefront, you can form an interference pattern in 3 dimensions. And so this is the microscope setup here. So there's a lot of bits, but the important bit is right here. So this is a spatial light modulator. The device itself is a liquid crystal that's actually not so different from what's in a computer screen. And it has a pattern that looks something like this, and a pattern that looks like this actually then produces an image like this in the sample plane. So this is the objective of the microscope, and this fish would sit right here. And so the computation I'll talk about briefly. So you have some desired intensity distribution, and you want to turn this into the pattern that will produce that. So there's this algorithm that exists called Gerkberg-Saxton that we use, and it involves the FFT because actually there's a duality for light that you can treat it either as a wave or as a series of angles, and so you can use the FFT to go back and forth between that, which I won't cover in detail, but there's a good talk on MIT OpenCourseware if you search Fourier 4F. So it then iteratively uses the FFT to go between the desired intensity distribution and the phase correction you would require for that, and then inverts it back to come up with a computed distribution. You have some error, and this iterates, and eventually you have a nice solution. And actually for this, we thought about implementing it in CUDA, but it turns out the Python version is sufficiently fast enough. And there's some caching on top of it, so it will only ever compute a pattern once. So if the pattern's re-requested later, it just pulls it out of the cache using joblib, which is quite effective. So using this, here we have a microscope objective, here's a sample, and rather than focusing the light into one point, now we can focus the light into multiple points simultaneously. So this is a close-up. So this is the axial dimension, so into the sample, and you can see that rather than sort of a single point of focus, we have two different points of focus, and we take sort of a top view we can control the light in 3 dimensions to form patterns as we need. So a little bit on the software side for this system. There's several different parts, and so I chose to implement a server-client model. So there's one holographic server that sits with the hardware and handles the calibration for that. And then there's multiple clients. So the microscope is one client because you need coordination between the imaging you're doing and the stimulation. You also need user interface, and different users could want different user interfaces. Like for some people that don't program, you want a graphical interface. For other people that like to program, they might want to very directly define the patterns just with pure Python. And so the system I used for that, for message passing, is ZMQ. Inside there's protobuf serialized messages and metadata, which I heard earlier the guy from DLR was saying they use something similar, so can't be so wrong. And to store the format, I wanted to use a vector format instead of a raster format for more precision. And so I just used the SVG format, which is great, because you can open up an SVG and easily read it. It's nice XML. And it's very precise, and you don't have to invent a bunch of new things because it's already there. For the user interface, I use Qt. And using Qt.py, you can very easily switch between PyQt or PySide, which is quite helpful. And essentially, the users can load in a recorded image volume. There's a calibration procedure to align that to the microscope's coordinate space. So when the users draw a pattern onto the image that was recorded, the holographic system knows where in its spatial coordinates it has to go to make the same pattern. And yeah, they can define the patterns directly or use SVG. And for handling the timing and duration, that was a little bit of a challenge because there's a lot of different possibilities of how to do that, and I wanted something that was both easy to use for people that don't like programming and very powerful. So the solution I went for is loadable Qt widgets. So there's a base class that you can inherit from, and then anything inheriting that base class can be loaded dynamically into the program. So then even as the program's running, I can change some parts of the algorithm for assigning the timing and reload it. But also there's graphical parts, so if someone wants just something very simple and purely graphical, that also fits in. So here's the application of the system. So for the imaging, we go into 3 different dimensions using a electrotunable lens. So it's a soft lens that by applying a voltage, you can very quickly change the shape of the lens. And we could use this to image several volumes per second through the fish brain. And so for instance, here's one plane through the brain. And we have several different neurons, and here's another plane, and we can pick out 2 of the different neurons, this in this plane and this in a plane further up. And we can do our imaging and stimulation. So what's here is an activity trace recorded from the neurons, and the yellow bars are when the stimulation is active, and in this case we targeted cell 13 for stimulation, and we can see that when we photoactivate, the neuron is excited and the activity increases, but not in the other neurons. And this is the SLM correction pattern that generates this distribution in space that corresponds to this neuron. We can also choose some other neuron, do something similar, different spatial pattern, different excitation, and also we only activate this neuron, or we can do multiple neurons simultaneously. So we can select both, and you can see actually that the pattern sort of looks like some weird mixture of the other patterns together, and we can activate these 2 neurons very precisely without affecting the rest of the circuit so much. But actually we want to affect the rest of the circuit, and we want to go in, stimulate multiple neurons, and figure out how the brain actually works. But this is a bit of a problem, because you could say, well, let's go in and try every different possible combination. But that's the number of neurons factorial, which is quite a problem, because the zebrafish has maybe 90,000 different neurons, and even in a small sub-circuit, even 100 factorial is a very, very large number. So in this case, we decide to use behavior to guide the stimulation process. So we start from larger regions, and so for instance, if we activate in region B, we find that the tail is activated and steers, and if we activate in region A, we don't see any activation. And from this, we can iteratively try smaller and smaller patterns with a sort of leave one out approach to find effective patterns for driving behavior. So in this case, we can try different combinations of 4, and then 3, and then 2, and use some simple regression models to, for instance, estimate that these cells contribute particularly strongly to the behavior compared to some of these other neurons in the circuit. And then ultimately, we can use this to find groups of behavioral neurons that when we stimulate them drive steering and swimming behaviors. At the same time, watch these behaviors and watch many other neurons in the circuit that are involved in the activity. So when there's red text up here, that's stimulation. You can see the tail moves. And for the first 2 trials, it's a small tail deflection. And then for this third trial here, you can see that there's more activity spread throughout the circuit, and that it's a much larger, like, full swimming behavior. So, how to do the analysis? This is quite a challenge, because looking at 500 neurons at once is very difficult, and there's a lot of different possible patterns. So one of the really useful tools for this is dimensionality reduction. So you can take dimensionality reduction, principal component analysis or independent component analysis, or even some of the more complicated versions, and apply it to capture common activity patterns throughout the network into a smaller number of components. So in this case, 3 components captures actually a reasonable amount of the variability in the circuit, and is a little bit easier to interpret. So you can see here that this bold component is active when the stimulation is active. So this includes, for instance, the neurons that are being stimulated and some of the surrounding neurons that are very highly linked to them. There's another group of neurons that's spread throughout the circuit much more broadly that's activated by the larger swimming behavior. And then there's some sort of slower changes in the network state. This is another problem, actually, is that sort of in a nice mathematical model, everything stays the same. But in a real-life brain, as you go in and start to poke stuff and activate neurons, other neurons adapt. And so you have to hit a moving target, which is an interesting challenge. Another useful approach is that we can use clustering. So for instance, to go in and look at the behavior. So sometimes when we have activations, we have small bending behaviors where the tail deflects. And other times we have larger swing behaviors. So in this case, it was, we used hierarchical clustering, which worked pretty well. But there's also many more complex methods. So for instance, there was a cool paper recently using autoregressive hidden Markov models in mouse behavior to look at sub-second timescales. And finally, we can do some modeling. So one of the questions we like to ask, and sort of to show that we actually can learn something about the brain, is to try to predict the behavioral output given the state of the neural circuit. So what we can do is, of course, split our data in half, because cross-validation is important. Train a model. In this case, it's actually quite simple. And then ask it, based on neural activity, can you predict what the tail is doing? And it works reasonably well. Not perfectly. You might not expect it to work perfectly because we're only looking at a small number of the neurons in the brain. So there might be other dynamics happening outside this area that we're missing. And if we use all of the neurons, we can get a reasonably good fit. But this is a little bit of a challenge in that there's so many neurons and we don't have that many trials, so we're actually massively overfitting to the data. So what is much more robust is to do something like reduce the space, because anyways a lot of neurons are highly correlated with each other, using independent component analysis, and then use these independent components to run the regression, and it performs slightly worse, but not so much, and it's a much simpler model. So challenges. The behavior is complex, and the neural circuits themselves are even more complex. In a similar way to that in a deep learning neural network, the output layer is usually the simplest one, and sort of everything comes together, and then some of those intermediate layers have very complex features. There's some really difficult multi-dimensional data sets, and a lot of the statistical properties that you would like to have, like normality and equal variances, are very rare in this area. And one of the biggest challenges when you're getting down to precise exploration is differences between individuals. So every zebrafish is slightly different from every other zebrafish, and every person is fairly different from each other person. And so when you go in there and look on fine detail, it's very hard to sort of map these features from one person or individual to the next one. And one of the final challenges is making tools and analysis that biologists can use without having extreme amounts of programming knowledge, and that are also transparent. Because it doesn't matter if you have the best, most accurate model in the world, if you can't explain it to other biologists, it won't be of much use to them. So I'd like to say thanks to Marco for from my lab who worked with me very closely on this project, and then also Thomas and the whole rest of the biolab. Contributors to the mini libraries used. All the figures are done in Matplotlib. There's skimage, or sklearn, NumPy, Python, of course, SciPy. It's very great because in Python you can just do everything. This is one of the reasons I think it's so great as a science language. We also have a database for the lab to keep track of our fish that runs in Django. Whereas compared to some of the other languages where they do one thing like statistics reasonably well, but you can't use them for many different things in the lab. And if you're interested, if you have questions, if you have suggestions on how to improve the analysis or data workflow, I'd love to hear it. Send me an email or come find me afterwards. Thank you.
Thank you very much, George. So, is there any questions? Come on.
Usually in an experiment, how much data do you collect, and how much does it time you take to analyze them?
It depends on the system. So like some of the simpler microscope systems we have aren't so bad, but we have a new one that can produce 500 megabytes of data per second. So that's a little bit of a challenge. And then usually we keep the fish in the microscope for several hours each, but oftentimes we give them breaks in between individual trials. So it can be a reasonable amount of data in that. So for a single fish, you could have 100 gigabytes of data. And then, of course, you want to do this across multiple fish, so the scale of the data can also be a challenge.
If there are no questions, then we will move to the last speaker of today. Thanks again.